you. Okay, so hello everyone. First of all, I would really like to thank Harry and Dylan for the invitation to this wonderful webinar series. I'm really happy to be here with you today. For those of you who might not know me, my name is Mateja, and I just recently started as a group leader at the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany. And today I will be talking to you about how we can use ancient genomes of Paleolithic humans and late Neanderthals to not only reconstruct their population history, but also to dive into their fine-scale interactions. So as most of you already know, modern humans first appeared in Africa at least 300,000 years ago. And somewhere between 100,000 and 60,000 years ago, a subset of those humans migrated out of Africa and spread across the world. At the same time, other hominin groups were present in Eurasia, such as Neandertals, here in blue in the West, and their sister group, the Denisovans, for whom we can only assume their geographical distribution, were present in the East. However, somewhere between 50 and 40,000 years ago, when modern humans started spreading across Europe, Neanderthals disappeared. And how these interactions and changes actually took place has long been debated. This time period is also known as the Middle to Upper Paleolithic transition or period, as it is characterized by a lot of changes in the archaeological records across the archaeological sites in Europe. So here in this map, it is summarized what was known at the time about the attribution and the distribution of so-called transitional archaeological assemblages or industries to those that were made by Upper Paleolithic humans and that are highlighted here in different colors, or the ones that were attributed to Neanderthals in different dashed lines. However, hominin remains which are directly associated with these transitional archaeological assemblages are extremely rare. Back in 2010, by sequencing the draft Neanderthal genome, it was shown that Neanderthals contributed around 2% of their ancestry to all present-day humans that live outside of sub-Saharan Africa. And based on actually the size of the segments of Neanderthal DNA and their distribution, found both in present day, but as well as ancient human genomes, it was determined that this main pulse of Neanderthal introgression, or the main pulse of Neanderthal ancestry coming into all present-day non-Africans, happened somewhere between 54 and 58,000 years ago, and most likely in the Near East, so before all of these groups of humans split from each other and spread across the world. However, where, when, and how often modern humans actually interacted with the local archaic groups, or in this talk we'll be focusing primarily on Neanderthals, was not that well understood. So in order to actually study the interactions between modern humans and Neanderthals in a greater detail, it would be great to actually study the genomes of the individuals that lived close both in time and in space when those groups could have met each other. And here it is plotted the distribution of all of the published ancient human genomes to date. that have been sequenced to various levels of completion. And here I'm only plotting the ancient humans. So in the recent years, there has been a big explosion in the number of individuals from whom genome-wide data has been retrieved. In November of last year, that number was well over 10,000, and this number is already outdated nowadays. However, if we move further back in time to only 30,000 years, and even including the wonderful data that has been published yesterday, we are down to only 30 individuals from 17 archaeological sites. And again, here are just the ancient humans and not the Neanderthal or Denisovan individuals that are plotted. So when we move further back in time, the number of individuals from whom we actually have the genomic data from drops down quite drastically. And if we go even further back in time to the modern humans that are older than 40,000 years, and that they themselves or their immediate ancestors could have met Neanderthals, up until recently, we only had 3 individuals from 3 archaeological sites from whom genome-wide data have been retrieved. Those are the 45,000-year-old Ust-Ishim individual from Siberia in present-day Russia, the approximately 40,000-year-old Tianyuan individual from China, and the 40,000-year-old Oasa-1 individual from the area of present-day Romania. And this is not only due to the scarcity of human remains from this particular time period I'm very interested in, but also due to the properties of ancient DNA and the preservation of the true endogenous DNA in temperate environments. So as most of you might already know, if we go to the permafrost environments, you can get ancient DNA that is up to 2 million years old. However, in the temperate environments, such as in Paleolithic in Europe, it's a bit more tricky. And this is due to the fact that after an organism dies, its DNA, which is here in these yellow-orange bands, starts degrading, becoming shorter and shorter over time, but also it accumulates certain modifications. At the same time, microorganisms and fungi that colonize the remains leave their own DNA behind. But for us working with ancient hominin remains, the biggest challenge represents actually the contamination which is introduced from present-day humans. And this contamination can stem from various sources and can be introduced at different levels of processing of the hominin remains. So it can be already introduced during the excavations of the post-handling of the remains, but also in the lab, so it can be introduced in the laboratory settings. So in order to minimize the levels of present-day human contamination that we could introduce in the lab when we are trying to get the precious DNA from the ancient specimens, all of the ancient DNA work is conducted in so-called clean room environments, and we are dressed up in these hazmat suits that most of you got to be familiar with, unfortunately, within the last 3 years. But this is not so that we protect ourselves from the specimens or from something that might be in them, but actually to protect the specimens and to protect the DNA from ourselves. And I mentioned already these certain modifications that happen in ancient DNA. And those modifications are actually some sort of a blessing, if you will, because there are certain modifications which are characteristic for ancient DNA and can help us distinguish true endogenous DNA from the contamination. Those modifications, which are characteristic, stem from the amination of DNA cytosine bases into RNA uracil bases And those uracil bases are actually recognized as timing bases by DNA polymerases that we use in the lab when we are copying molecules of DNA and creating so-called DNA libraries that I will explain to you in a little bit. And so these changes or modifications that you might have seen in ancient DNA papers are called so-called C-to-T changes or cytosine to timing substitutions. And so these modifications are especially prevalent or represented in higher frequency at the ends of the true autochthonous DNA molecules stemming from your specimen. And they're almost completely absent in present-day contamination. So you can use these modifications to enrich for your endogenous DNA, either in silico on the computer, to differentiate between the ancient DNA from your specimen and the more recent contaminant, or directly in the lab. So you can actually enrich or fish out the molecules that have these uracil bases in the lab, and in that way try to reduce the levels of present-day contamination. Another characteristic which might be useful for the downstream analysis in some cases is that ancient DNA is usually much shorter than present-day contamination. So you can also use the size filtering to try to boost your endogenous DNA or the proportion of sequences that are stemming from your specimen of interest. And here is just a brief overview of the ancient DNA lab workflow for those of you who might not be familiar and how this process actually goes. So for me, I usually start with bones or fragments of bones or teeth. And in the last few years, a lot of people have also been working with sediments as a primary source of ancient DNA. But in cases where we do have bones or teeth, what we first do, we drill small amounts of powder from which we then extract DNA using different enzymes and buffers to actually release the DNA from the powder out and digest the actual bone or tooth powder. Then we take small amounts of that DNA extract, which is now the soup of molecules of different sources that I showed to you in the beginning. So you have a mix of everything. And to these DNA molecules, in the step of what we call library preparation or constructing of DNA libraries, we add adapters to the ends. And because the amounts of DNA we have in the ancient specimens is so small, what we usually need to do is amplify that DNA and tag it with specific different sets of oligos at the ends. And after that, you can either sequence your DNA libraries directly on a sequencer, or what you can do is you can enrich DNA libraries for certain regions in the genome that are of your interest, and then sequence them after that. And this is just an illustration of the nuclear DNA content we have in different ancient specimens. So for the 2010 paper, which was the first draft Neanderthal genome, was actually a composite genome from 3 different bones of Windia Neanderthals. The proportion here in yellow of the sequences of the nuclear DNA that we can align to the human genome, which some of the people then called endogenous DNA, was around 4%, and the majority of the other sequences were actually of microbial origin. In the case of our second high coverage Neanderthal genome, which we published in 2017, this proportion was around 10%. And in the case of the first high coverage Neanderthal genome, and what almost never happens with the specimens that I actually work on, and it's really in exceptional cases in the Paleolithic that you have this, such as in the Denisova cave in Russia, is that you have this proportion up to 70%. So most of the specimens that I work with look like this. So the proportion of sequences aligning to the human genome are at most 1%, and sometimes the majority of those sequences can stem from present-day human contamination. So how do we actually go about this? So for the cases where we have very small amounts of endogenous DNA, we use these enrichment strategies. So as I said, we fish out the molecules from the soup of molecules you have in your DNA libraries for specific sites in the genome, which are informative about the relationship of individual you want to study to other present day or ancient humans, but also Neanderthals and Denisovans. And depending on how much of this yellow proportion you have here, you can actually sequence your libraries directly to obtain either low coverage or high coverage genomes. And there is a lot of debate, what is a high coverage genome, what is a low coverage genome? And usually what it means is that you have each site in your genome represented by a few sequences, DNA sequences in case of the low coverage genomes, and 20 or more sequences are covering each site in the genome in case of the high coverage genomes. And for the in solution hybridization capture, this is just an illustration. So where you have your soup of molecules in your DNA libraries, you use these synthetic probes, which are targeting the sites in the genome you are interested in to actually enrich your libraries for those specific set of sites that you then sequence. And this way you are making the specimens that have very little of the endogenous DNA, but a lot of, for instance, microbial contamination accessible for downstream analysis. And actually those approaches are one of the main drivers behind this explosion of genomes of ancient individuals in the recent years. So recently, the number of individuals that are older than 40,000 years of ancient humans was increased from 3 to 8 by obtaining the genome-wide data from additional individual called Zlatý Kun from Czechia, which is around 45,000 years old. But what I will be focusing on for the purposes of this talk, a part of this talk, are the individuals which have been directly radiocarbon dated to around 45,000 years before present from the site of Bacho Kiro cave in Bulgaria, and which have been directly associated with the initial Upper Paleolithic archaeological assemblage. So as I mentioned already, Bacho Kiro cave is a cave in present-day Bulgaria, and it has been excavated, the things I will be talking about today, between 2015 and 2018. And a number of hominin remains have been found along with one human tooth in the layer I, which is the layer which contains this initial Upper Paleolithic archaeological assemblage, and which have been directly radiocarbon dated to around 45,000 cal BP. And there are two additional individuals which were found further up in the stratigraphy in the main sector area of the cave that has been excavated, and which have been dated to around 35,000 years cal BP, and which are most likely associated with the Orignation archaeological assemblage. So I have been really lucky to work with a team that has been excavating the Bacho Kiro cave. And what we did is, first we managed to obtain the full mitochondrial genomes of these individuals and show that they actually are ancient modern humans. However, to fully understand because they are 45,000 years old, and they fall within the time when we still have Neanderthals in Europe, it is also interesting for us to look at their nuclear DNA to see what about the interactions with Neanderthals and if these individuals had a chance to actually meet the Neanderthals. One of the first things which we were interested in is how these initial Upper Paleolithic individuals are related to present-day humans and ancient modern humans. And we can do this by comparing the genomes of these individuals to present-day and ancient human genomes. And here we do it with a form of a statistics where we look how much of the alleles does our individual that we are interested in share with other groups of humans. So here, just for illustration, I'm showing the sharing of these alleles. So how much this individual is sharing with a given present-day human population or with a pre-agricultural hunter-gatherer from Europe. So in this case, it is approximately 38,000-year-old Western Eurasian called Kostenki 14. And in the case of the previously sequenced 45,000-year-old individual called Ust-Ishim from Siberia that I showed you before, what was interesting, and the same was the case for the 40,000-year-old Wasa-1 individual from Romania, that it doesn't seem to have contributed much or detectably to the later populations. However, when we looked at the individuals from the Bacho Kiro cave, the initial Upper Paleolithic individuals, what was surprising is that actually they shared significantly more of their alleles with the present-day populations from East Asia, from the Americas, Central Asia, and Siberia than what they share with the later populations, which are in Europe in the Upper Paleolithic. So if we then compare a suite of our ancient individuals to the individual from West Eurasia, which is already on the lineage to the later Europeans, such as Kostenki 14, or the early East Asian individual, the 40,000-year-old Tian Yuan from China, we see that our initial Upper Paleolithic Bacho Kiro cave individuals are significantly closer to the ancient East Asian individual than to the later West Eurasian individuals, similar to what we see for some of the ancient American populations or some of the Central Asian and Siberian populations. And it is not only the case of sharing these alleles with Tian Yuan, but when we compare the initial Upper Paleolithic individuals to a set of other ancient human genomes, a similar picture emerges. So our initial Upper Paleolithic individuals from Bulgaria share significantly more alleles with other ancient East Asians, Native Americans, and Central Asians and Siberians than they do with the later West Eurasians. However, they do not completely disappear, or their ancestry doesn't completely disappear from parts of the Upper Paleolithic Europe. So what we have seen back in almost 2 years ago was that this ancestry survives in some form in approximately 35,000-year-old individual from Belgium associated with origination, or for those of you who might already have seen the paper that came out yesterday, also in some of the individuals that are around 23,000-year-old in Iberia. So they are not completely gone. And this, for me, it was surprising. It was not as surprising for our collaborators that we worked with on the Bacho Kiro cave remains. As I mentioned before, the Bacho Kiro cave individuals are directly associated with the initial Upper Paleolithic assemblage, and the sites with the initial Upper Paleolithic assemblages are found from East Europe to Mongolia or present-day China. Therefore, we can hypothesize that at some point this ancestry disappears or is diminished in the West Eurasia, but it survives in some form in the later East Asians and subsequently then Native Americans. But as I said before, what we were particularly interested in was the interactions between Neanderthals and modern humans. And just for illustration purposes, if you imagine that one of your parents is a Neanderthal, which is illustrated here in blue, and the other parent is a modern human, then an offspring of these two parents will have half of his genome coming from a Neanderthal and the other half coming from a modern human. However, if that child then continues on living in the human societies, the DNA from Neanderthals will break down over time into smaller and smaller chunks and will be present in these small DNA chunks, which we can find in people living today. Therefore, we can reverse engineer this so we can look at the actual sizes and distribution of these Neanderthal segments in the genomes of our ancient individuals, and then use them to date the time of the admixture between Neanderthals and modern humans. So if we look at the genome-wide distribution of Neanderthal ancestry in the 45,000-year-old individuals we have from Czechia and from Russia, this is how the distribution of Neanderthal ancestry looks like in the genomes of these individuals. And based on the sizes of the Neanderthal DNA in their genome, we can actually date the admixture that has happened somewhere between 80 to 100 generations before these individuals have lived, so a few thousand years before they were actually around, so in their tree, family tree. Previously, back in 2015, when we obtained genome-wide data of OASA1, the big surprise was finding these extremely long Neanderthal chunks or segments of Neanderthal DNA in his genome. And when we looked at the distribution of Neanderthal DNA in our initial Upper Paleolithic Bacho Kiro cave individuals, at least for me, it was surprising we found it again. So what are the chances that we find it again in the modern humans in Europe that actually overlapped in time and space with Neanderthals? And we did not only find this in one, but actually in all three, or indeed 4 individuals of the initial Upper Paleolithic from Bacho Kiro cave. And when we then use these segments to try to date the time to the Neanderthal ancestor, we actually see that it's just a few generations before these individuals have lived. So we can distinguish the main pulse of Neanderthal ancestry from the more recent pulse in the family history of these individuals. So if you look at the actual distribution of these Neanderthal segments, you can see these 2 slopes and actually distinguish these 2 pulses. So in case of the molar from the Bacho Kiro cave or the OASA1 individual, these individuals had Neanderthal ancestor 4, 5, or 6 generations before they actually lived. And if we use the human generation time and calculate like how far back in time that would be, it's just around 200 years before these individuals were living. And these 2 recent pulses we find in Bacho Kiro cave individuals and in OASA1 are distinct. So there are at least 3 separate pulses of Neanderthal contribution to modern humans. Therefore, so this is just summing it up. The main pulse, which most of the present-day non-Africans are sharing, and then additional admixture events with Neanderthals. And given the amounts of Neanderthal ancestry in the present day and ancient East Asians, it has been argued of additional pulses or dilution in the West. And up to today, these are the Neanderthal genomes we have. So we have 3 high coverage, high quality Neanderthal genomes sequenced. More are actually underway. The first one comes from 130,000-year-old Alta Neanderthal from the Denisova cave in Russia. The second one comes from approximately 45,000-year-old Vindian Neanderthal from Croatia. And the third one is from an 80,000-year-old Chagirskaya Neanderthal in close proximity of the Denisova cave. And we have a number of low coverage Neanderthal genomes. And these genomes are actually quite important, not only in our understanding of the population history and the dynamics between these different Neanderthal groups, but also in looking in the interactions between Neanderthals and modern humans, and in trying to disentangle which of these Neanderthal populations would actually be the closest one to the one or ones that have contributed their ancestry to the ancestors of all present-day non-Africans. So if we construct a neighbor joining tree of the nuclear genomes of our late Neanderthals and the 130,000-year-old Alta Neanderthal from the Denisova cave and approximately 90,000-year-old Mizmaiskaya 1 in the Caucasus in Russia as well. What we actually saw at the time that the relationships between different Neanderthal populations closely correspond both to their temporal but also their geographical proximity. So the 2 Neanderthals from whom we have genome-wide data from Belgium have in all tests been significantly closer to each other genetically than they are to other late Neanderthals or earlier Neanderthals. The Neanderthal from France, from Le Coté, again shares in turn more alleles with the Neanderthals from Belgium than it does with the Neanderthal, for instance, from the Caucasus. However, what we've seen as well is this temporal aspect where we have 2 Neanderthals which come from exactly the same cave in the Caucasus, but the later Neanderthal, the 45,000 Mizmaiskaya 2, which is not surprising, is significantly closer to other late Neanderthals than it is to Mizmaiskaya 1 that lived in that cave around 90,000 years ago. And as I said, we can then use these genomes to try to see which of these Neanderthal populations or groups would be the closest one to the one that we find in the genomes of all present-day and ancient modern humans. And if we look at, again, the sharing of the alleles or genetic proximity to the introgress fragments we find in present-day genomes and ancient genomes, we can actually pin down when this Neanderthal population that contributed its genes to modern humans formed. So on the tree relating the genomes that are also here, but also adding the high coverage genome of an 80,000-year-old Chagirskaya 8 individual, we see that actually this Neanderthal population formed somewhere here, as we see that the Neanderthals that we observe in the genomes of present-day and ancient humans are significantly closer to the later Neanderthals than they are to the earlier Neanderthals. And based on the split times between these genomes, we know that this Neanderthal population must have formed somewhere between 90 and 130,000 years ago. However, where this population was, it's still a while to go. And there was a bit of a discrepancy already with looking at the nuclear DNA versus the mitochondrial DNA of Neanderthals, modern humans, and Denisovans. As you see in the nuclear DNA, Neanderthals and Denisovans are actually sister groups, and modern humans are outside of the variation of the archaics. Whereas if we look at the mitochondrial DNA of all of the published Neanderthals we have to date, Denisovans, present day and ancient modern humans, individual from Cima de los Huesos, and using a chimpanzee as an outgroup, actually Neanderthals and modern humans are closer to each other, and then Denisovans are an outgroup. Also, the time to the most recent common ancestor was actually not fitting between the mitochondrial DNA and the nuclear DNA. And at some point, as we observed, quite disappointingly, we were mostly getting good nuclear data from, for whatever reason, female Neanderthals. So we went back to the lab to try to obtain Y chromosome sequences of some of the Neanderthals and Denisovans we managed to identify by generating these special targeted captures. So to fish out and try to generate Y chromosome sequences from the known few male Neanderthals and Denisovans that we had. And this work was done by Martin Peter. And again, on the Y chromosome, what we see is very similar to mitochondrial DNA, that actually Neanderthals are closer to modern humans and Denisovans are an outgroup. And the current hypothesis that we are working with is that an early contact between Neanderthals and modern ancestors of modern humans, which then introduced both the mitochondrial and Y chromosomes into the later Neanderthals and completely replaced the previous Y chromosome and the mitochondrial DNA of Neanderthals. However, what I'm particularly interested in is we are now continuing finding early humans among those groups of early human dispersals into Eurasia that have very recent Neanderthal ancestry, and it keeps on happening over and over again. What about the late Neanderthals? So the Neanderthals that actually would fall within the time frame when modern humans are already in Europe and from whom we have genome-wide data from. So if we look at the genome of one of the late Neanderthals, where we have a really good quality data from, and which is approximately around the same age as our modern humans from the Bacho Kiro cave, we actually do not see any introgressed modern human DNA in the genomes of late Neanderthals. And this is the case for all of the late Neanderthals that we have looked so far and that actually post-date the modern human arrival into Europe, which is actually, at least for me, really interesting. So I hope that with this talk, I managed to show you that ancient genomes provide us with a lot of information about past events that would not be so easily accessible with analyzing present-day human genomes alone. Even though present-day genomes contain a lot of information about past processes that have shaped them and formed them, it is really hard to disentangle some of the fine-scale things which were happening in the past and that have been obscured by later admixture of different human groups or migrations and turnovers and disappearance of some of the human groups from the genetic record. And these ancient genomes actually allow us to test certain hypotheses directly, but I would argue that ancient DNA research and ancient genomes cannot live on their own without an actual collaborative approach with archaeologists and paleoanthropologists. So it's just an additional layer of information, but it should not and never stand on its own without information from the archaeology and paleoanthropology. And based on the genomes that I showed you today and more that are actually underway, there is a complex history of Neandertal-human interactions, as we are seeing that this introgress Neanderthal population formed between 90 and 130,000 years ago. There are multiple pulses of Neanderthal admixture that are independent of each other. And the admixture between Neanderthals and modern humans keeps on occurring when they meet. And this early contact between the ancestors of modern humans and early Neanderthals actually led to a complete replacement of Neanderthal mitochondrial and Y chromosome lineages. However, in the later times, when we have the migration of modern humans out of Africa, and the overlap of modern humans and Neanderthals in Europe for several thousands of years. For the Neanderthals we have looked so far, we cannot find any evidence of a recent modern human gene flow into them. So it's either a bias in our sampling, or it's just a matter of time until we find a Neanderthal with a more recent human gene flow. And with this, I would like to thank our collaborators without whom this research would not be possible, as well as the funding bodies. And I would like to thank you for your attention, and I'd be happy to answer any questions that you might have.
Thank you very much for that, Mateja. That was really insightful, especially for those of us that perhaps don't spend enough time with this sort of data that we really should. It's obvious yet again that we can't have anything simple in this field ever. So really insightful. Thank you so much for that. I have noticed that we do have some questions in the chat, so I'll go to those first. But for anyone that has a question, if you'd like to stick your hand up, I'll come to you once we've asked the chat questions. So the first one is, it's from Walter. Has genomic research shed any light on why Neanderthals or Denisovans became extinct?
It's a great question. I wish I would be able to answer simply to this. There are probably multiple factors which have led to the disappearance of Neanderthals and modern humans. What we can say from the increasing the number of genomes of ancient humans and also Neanderthals is that Neanderthals actually had a very small population sizes. So their effective population sizes were quite small, but they were quite small for quite a long time. The other thing is that we are seeing these multiple occurrences of Neandertal-human admixture with Neanderthal contribution to the early Upper Paleolithic humans. So one of the hypotheses could also be that some of these small Neanderthal groups that were in Europe actually got absorbed into the bigger human populations that were migrating into their territory. But I think it's far from a definite answer, and there might be a lot of different things which have contributed to their disappearance. Some of the people have argued that Neanderthals and Denisovans haven't really disappeared as they continue to live on in our genomes. So actually, if we would take on all the Neanderthal DNA in the genomes of humans living today, it would be way more Neanderthals than has ever been during the time when they were actually around. But yes, it's a hard question. One of the interesting bits is that also these early groups of humans, so populations to which Ust-Ishim, Oase, or Zlatikun, or even Bacho Kiro cave individuals have belonged to, disappear as well. So, or their genetic ancestry, they don't contribute much, or some of them at all, to the later groups that we find in Europe. So it's not just that Neanderthals disappear, but also these early groups of humans. So it's a complex thing, I'm sure.
Thanks for that. Thanks, Walter, for that question. We do actually have another question from Walter.
Yeah, sure.
Are genomic changes and cultural innovations coupled?
That's a great question. So in certain cases, where we see the changes in the archaeological assemblages, and now I'm talking about modern humans during the Upper Paleolithic, we see also the changes in the genetic ancestry. So we have different genetic ancestries which are coupled with different stone tool industries. However, it's not always the case. So we, especially the big paper that just came out yesterday, in case of Gravettian, where there are some slight cultural changes, but I'm not an archaeologist, so I stay out of these differences. We do actually see two different genetic components. So the one in the east and the one in the west. And the one in the east actually disappears, whereas the one in the west actually survives throughout the Ice Age and then contributes to the later Magdalenian individuals. And we had the other things as well, where we had genomic data of back in 2016 of these Eastern Gravettian individuals, and then individuals from around the Lake Baikal, such as Malta and Fontevagora, which produce similar types of figurines, these Venus figurines. However, again, their genetic ancestry is completely different, and like even more different than we had between the Western and Eastern Gravettians. So I think it could be that actually in some cases we have the same culture by different populations, but also it could be the other way around, that the different types of stone tools or culture is produced by the same population.
Very, very exciting. Thank you. And thanks, Walter, for that question as well. We do have a question from Chris Stringer. He says that was a great talk. Are we any further towards finding out whether, and I'm assuming he means Neanderthal here.
Yes.
Are we any further towards finding out whether Neanderthal and Denisovan introgression came mainly from males, females, or about equally?
Yes. So it's really hard to say, given that in present day, but also in ancient humans, we do not detect any mitochondrial lineages from Neanderthals in modern humans, also in these early groups, and also no Neanderthal or Denisovan Y chromosomes. So but also the survival of certain lineages in modern humans, or the other way around, could simply be due to drift, so that they're lost or pruned away. So at this point in time, I'm not really sure we're close to finding out if it's coming through just males, just females, or both.
Thank you for that. And thanks for that question, Chris. That was a great question, actually. Great. I know that Walter in the chat has also asked for a reference title, journal, page of the big published paper yesterday.
So it is a paper published. So there are two papers that were published simultaneously. One is in Nature by Post and Yu et al. It's paleogenomics of, now I will butcher the title, Upper Paleolithic to Neolithic hunter-gatherers. And the other one was published by Vanessa Villalba in the Nature, Ecology and Evolution, and it's about the Solutrean individuals from Iberia.
Great, excellent. I see someone has actually also put the link in the chat for the so thanks for doing that as well. Excellent. Do we have any more questions at all? I am conscious of time, but if we have any more, that would be great. I'm sure we can fit one more in. If not, I think we'll say thank you very much for that, Mateja. Thanks. Really, really insightful, really lovely presentation as well. Thanks very much. And thanks everyone for coming. And I hope we'll all see you all again.
Thank you for having me. Bye.